This podcast is for informational and educational purposes only. It is not medical care or advice. Clinicians should rely on their own medical judgments when advising their patients. Patients in need of medical care should consult their personal care provider. Welcome to That's Pediatrics, where we sit down with physicians, scientists, and experts to discuss the latest discoveries and innovations in pediatric healthcare.
From UPMC Children's Hospital of Pittsburgh, this is That's Pediatrics. I'm your co-host, Amanda Pahalik, Assistant Professor of Pediatrics and Immunology.
And I'm your co-host, Arvind Srinath, Associate Professor in Pediatric Gastroenterology.
Today, our guest is Dr. Guang Li to talk to us about his research on growing a four-heart chamber in a dish. Dr. Li received his PhD training in the Chinese Academy of Sciences in Shanghai by studying the epigenetic regulation of plant organ development. After that, he joined the Stanford Cardiovascular Institute as a postdoc to study heart development in mouse and human iPSC systems, and by combining single-cell mRNA sequencing, single molecular in situ hybridization, machine learning, and advanced imaging techniques, he systematically analyzed the temporal and spatial development of the heart at the single-cell level. In 2019, Dr. Li came to UPMC Children's Hospital to start his own lab, where he continues to use high-throughput technologies to answer questions about heart development as it's a critical embryonic developmental process. Thank you for being here. So tell us a little bit about your path to UPMC Children's Hospital and how you became interested in studying the development of the heart.
Oh, thank you so much, Amanda. Thank you for having me. So it's my great honor to be here. So I actually started here 3 years ago. Before coming to Pittsburgh, I was an instructor at Stanford University, as you have introduced. So I did 5 years of postdoc training and 1.5 years of instructor there. So I was always, I would say, interested in the development of biology since I started, to take the biology major in college. I started plant development in my PhD, as you have mentioned as well. So I studied the epigenetic regulation of the plant morphogenesis, back then. But after graduation, I start to think about, to do something more related to medicine. Mm-hmm. So that's why I joined Stanford Cardiovascular Institute, Dr. Shang Wu's lab, to study heart development. And, after the 6+ years of training there, so I think I have been tightly related to the heart development field.
So from a clinician standpoint, here's a really basic question, but how do you study the developing heart?
Yeah, we use 2 different models. We use mice, and use human-induced pluripotent stem cells, as models. So for the mice, we have different types of mice. Either have the genetic mutation that can cause the heart developmental defects, or have those chance genes, the reporter genes that can lineage, can label, a specific cell type and trace the lineage, across the whole developmental progression. So we basically use the combination of the 2 types of mice to understand the molecular and the cellular, mechanisms during the heart development. And we also use another model, human-induced pluripotent stem cells, or called human iPSC. So those cells can be generated, from the human blood cells or human skin, fibroblasts, for example. And those iPS cells, or stem cells, they have the potential to lineage specify into any cell types in the heart. So then we can use those cells and to study the heart development in human.
Are you studying these heart cells via biopsy or post-mortem?
We use the biopsy samples, but we usually just draw blood, a tube of blood from the patients. That will be also okay. But it depends on the source, right? Blood cells or skin fibroblasts.
Got it. Thank you.
So obviously, heart development is important in the context of a children's hospital. So can you tell us a little bit about, as you mentioned, sort of the impact on human health? So how are children impacted by heart development when it goes awry? And what percentage of children are born with heart development issues?
Yeah, if the heart development does not go well, it can cause congenital heart defects. Congenital heart defects accounts for about 1 to 2% of all live births in the U.S. and in the world. So, the congenital heart defects can be, mild or severe. For those mild congenital heart defects, for example, there's holes in the septum, they can heal by themselves. They may heal by themselves, so there's no need of any intervention. But for some, severe types of congenital heart defects, they need to be repaired by surgery. Or there are some severe defects, they're just too severe to be fixed by surgery, or they can be fixed by surgery, but the heart will just function for a short time and then end up as heart failure. So for those severe types of congenital heart defects, more research are required to understand the mechanisms in the heart development to help develop, further or better treatments.
Okay, so that really brings me to my next question, which is, as a fellow researcher trying to sort of find the way to do research in the lab that's going to impact human health, what are the big gaps in our knowledge of heart development, and how is your lab working to close those gaps?
Yeah, that's a great question. I have to say I ask myself, all the time. So I think, as I said, we use two different models, mice and human iPS cells. So in mouse heart development, after those, I will say, decades of research in this animal, in this model, we already know those big concepts. We have accumulated a lot of knowledge, in those big stages. So I think that the gap now we have is how to connect those things, and to understand how the cells at early stage have developed all the way to a functional organ. So that's one gap. I think another gap is, we really do not know how much, we really do not know much about the heart development in human. Most knowledge actually come from mice, or other animals such as zebrafish, So this is another gap. So in my lab, we basically, to close the first gap, we are using those omics, single cell omics or single cell transcriptomics, epigenomics, those approaches to understand the whole picture of the heart development, across all the stages. Basically to understand the biology in every single cell across all the stages. So that's our first, try or approach. And in human, as I said, we are using human iPS cells, to lineage differentiate pluripotent stem cell into different cardiac cells in a 2D or 3-dimensional, differentiation system to understand the differentiation process in human.
Can you go into a little bit of detail with these omics that you bring about in terms of how is that technology used and what does it entail?
Yeah. So those omics actually have been advanced so much recently. Okay. We currently can go to the single cell level to understand the transcriptomics, meaning the transcriptional, genes expression.
Got it.
Called mRNA, right? Yeah. A level in the cell across the whole genome genes, in the single cell, and we can also understand the epigenomics. It means the DNA modification, or histone modification, in the cell. And we can also understand the even proteomics, meaning the translated protein in this single cell, at different stage. And now we have tools to map those omics data back to their in situ original, spatial location. So this is called spatial omics. So all those omics tools actually have combined, can really give us an idea, how the cells actually how the cells actually function together, to guide the cells at early stage all the way to the for fully functional heart.
Got it. Thank you for clarifying. And then looking forward, what clinical implications do you see moving forward from your work?
Trying to understand the genes, how they express, how they function, And that really need to relate to the congenital heart defects. For example, the genes we have identified that highly expressed in certain cell types turns out to be associated or tightly associated with the congenital heart defects. So that can really give us an insight, how this disease have been caused, in what kind of a cell type, by what kind of genes, at what kind of stage.
Got it. Thank you for clarifying. Appreciate that.
So once you have this, part of it is information gathering, right? You have these omics tools that help you at the single cell level understand, okay, let's see within the developing heart, every cell, what genes are expressed, what proteins are expressed from those genes, and where are they as the cells differentiate. Can you tell us sort of, even if it's a bit speculative, how that information then translates into you being able to actually consider the idea of growing a heart in a dish and what implications that has? So we grow a heart, let's, obviously this is a bit like the big goals to be able to grow the heart in the dish, but then once you've done it, like what will that mean for our understanding of heart development and for potential therapeutic options for children and for patients?
Yeah, so that's a very important question. A very important goal of our research in mice is to translate it into human. So I think one important application is to use the knowledge, right? If you can regrow a heart in this, that will be a very good demonstration. So currently, actually, we have mainly used the growth factors. We have identified, so many labs have contributed. Identified the growth factors, that function in mouse heart development. So we are using the knowledge in mice to lineage, differentiate the iPS cells into heart cells. So we use a system called organoid. So organoid is a self-aggregate construct with in vivo-like features.
Okay.
So the anatomy, is kind of like the in vivo heart.
Okay.
So we can differentiate or generate, atrial or ventricular heart organoids. So as you may know, heart has 4 chambers, 2 atrials, 2 ventricles. We think, if just one chamber, that's not, like the in vivo, right? So we need a method to generate 4 chamber structures. So I think there are two approaches to do that. One approach is to induce both atrial and ventricle organoids simultaneously or at once. But that's very challenging because it turns out atrial and ventricular need different growth factors. And how can you actually spatially separate them, in the dish?
Got it.
So the second approach is to generate atrial and ventricular organoids separately, and then fuse them later. So that's the approach we took. And we can, generate atrial and ventricular and put them together and form at least 2 chamber or 4 chamber, organoids. But that's just the first step. What we need to do next is to add the other structures that's called lung chamber structure, such as valves, septums, conduction cells. All those have been well studied in mice, and we know those growth factors, and we have protocols to generate them separately.
Wow.
And what we need to do is to add them together next, either by fusion or figure out some way to lineage specify them all at once.
No, so just to clarify, so at this point you can kind of build half a heart in a dish with two of the 4 chambers, and you can separately get a lot of the pieces of the heart. And so now the question is, how do we kind of make them all come together to be a single functioning unit?
Yes.
The heart.
Yes. We can generate, one atrial, one ventricular, and we can put about one atrial, one ventricular together, but without a valve. So we needed to induce the valves next.
And these all, and they all beat even?
They all beat.
That's incredible.
Wow.
So
Wow.
and this is from mouse cells.
Human iPS cells.
Human IBS cells. I see.
Yeah.
I guess I'm just curious, do they grow to the correct size even? Is there a way to,
Oh,
because the growth of an organ is also highly
Yes.
controlled, right? And
Yes.
so how do you make sure that the size of the organ, even if you've only got half of it, is the correct size, et cetera?
Exactly. The size actually can be controlled by some growth factors, and you can add them to make them grow bigger, or you can, add the inhibitors to reduce the size. But if you need to generate a size match with the in vivo hearts, so then we need to figure out what's the right, time points, time period, and what's the right dosage of those growth factors.
Wow. Okay. Yeah. So, and then I guess, can you share a little bit with us, like kind of what the sort of like in your mind, that big picture dream goal that we all have as scientists is, and like, like what is it going to take to achieve that? And then, once you have it, what will that mean for your field?
You mean the 4-chamber heart?
Yeah.
Dish?
Yeah.
I think there is still a long way to go, I have to say, because the heart is looks simple, but still actually quite complicated. Yeah. They beat does not mean they're actually really fully functional, right? So we need a really real size of lumen. The heart, the main function of the heart is to beat, to pump blood to the whole body, right? So the lumen structure will be another critical. And we need all the other types of cells, such as fibroblasts, endothelial cells, or conduction cells. This is still a long way to go. But once it succeeds, if we really have a fully functional, not fully, partially functional even, 4-chamber heart, that will be huge. Because we can use that to model those all different types of congenital heart defects, how they actually have developed on the cellular level. And if we introduce a mutation that come from those patients, let's say associated with the patients, and then see what kind of defects they could lead. And once we have those models, and the next step is to use them to screen, drugs, right? To identify the potential medicine, to treat those defects. So that will be, I will say, the two main, applications we can have.
So you're looking at prevention, which is fantastic. You're looking at prevention of these congenital heart defects as a long-term goal. Yeah. Is that following an assumption that they're all due to a gene mutation, or is there a subset which aren't due to a gene mutation in terms of congenital heart defects?
Yeah, that's a good question. So I think that they are both, right? Part of the congenital defects are caused by genetic mutations, and some are not, probably. Right. Probably contributed by, other factors, such as glucose or alcohol, all those different factors. I think once we have this model, we can model both of those causes, right? Either genetic or non-genetic factors. Yeah.
Thank you for clarifying. That's really interesting.
Yeah. Thank you. I guess in the last few minutes, I kind of want to ask maybe, sort of what are the current challenges that your field faces, or that you feel, sort of is a limitation of what you could really like to see achieved? And then maybe as a follow-up, like what do you hope to discover in the next 5 years?
Mm-hmm. So I think the limitation is really to connect, as I mentioned, I think it's really to connect those pieces. Another thing is to use to study the heart development in the human cells. Use the human iPSC or human organoid system. I think what I want to achieve, next is to further optimize, the organoid system to make the 4-chamber hearts actually as close as we can, right, to the in vivo hearts. That will be one goal. But another goal is we still need to continue the study using the mouse models, because our goal, I think, is to understand the biology in every single cell, temporally and spatially, across the whole developmental progression.
Wow.
Yeah, those are lofty goals, but I feel confident that you will achieve them.
Oh, thank you so much.
Thank you so much for sharing your research with us today and for taking the time out of your busy schedule to be here and talk to us about your science. We really appreciate it.
Thank you so much.
I echo Amanda. Thank you so much. This was fascinating, and I really appreciate you explaining what the amazing endeavors you're doing. Thank you.
Thank you. I appreciate it.
You can find other episodes of That's Pediatrics on Apple Podcasts, Google Podcasts, Spotify, and YouTube. For more information about this podcast or our guests, please visit chp.edu/that'spediatrics. If you've enjoyed this episode, please be sure to rate, review, and subscribe to keep up with our new content. You can also email us at podcast.upmc@gmail.com with any feedback or ideas for topics you'd like our experts to cover on future episodes. Thank you again for listening to That's Pediatrics. Tune in next time.